today's lecture we will continue the second part of the lymphoid tissue. Starting with the tonsils, they are lymphoid tissue that related to the mucosa-associated lymphoid tissue, and they are found in the initial portion of the gastrointestinal tract, and they belong to the mucosa-associated lymphoid tissue, so they are incompletely encapsulated, and they are of three types depending on their location. First, pharyngeal tonsils, then we have palatine tonsils and the lingual tonsils. The palatine tonsils, they are paired structures located at the lateral wall of the oropharynx, and they are lined, that means covered, by stratified squamous epithelium. This epithelium dips deeply inside the tonsils, forming a structure called the crypt, which are about 10 to 20 in number in each tonsils. In case of tonsillitis, this crypt will be filled by inflammatory cells as lymphocyte and bacteria, and also by desequamated epithelial cells. The lymphoid tissue in the tonsils is in form of a free lymphoid tissue and in form of lymphoid nodule with germinal centers in case of activation. The capsule is a band of dense connective tissue. It's located beneath the tonsils and separated from the underlying structure and prevent the spread of infection. In this figure, we can see this section in the tonsil. This is the lining epithelium. It is a type of stratified squamous epithelium, and this downward invagination is the crypt, as it's shown in this figure. This is the crypt filled with bacteria and desequamated epithelial cells. The lymphoid tissue in form of nodules with germinal centers and in form of a free band of lymphoid tissue. On the side of the capsule, it is a dense band of connective tissue that located beneath the tonsils, separated from underlying structures. The pharyngeal tonsils, it is a single located in the superior posterior part of the pharynx, and it is lined by respiratory epithelium, which is a ciliated pseudostratified columnar epithelium, and it contains no crypts. The lymphoid tissue in form of free lymphoid tissue and lymphoid nodules. It has no crypts. This is an important feature. The capsule is thinner than that of the palatine tonsils. A clinical condition called adenoid, which is seen in pediatric age groups, characterized by hypertrophy of pharyngeal tonsils, and sometimes it's block the airways. Lingual tonsils, they are numerous, located at the base of the tongue, in the posterior third of the tongue, lined by stratified squamous epithelium. Each one have a single crypt. Next subject is the thymus, which is a primary lymphoid tissue, as we discussed in earlier lecture. It is a type of lymphoepithelial organ located in the mediastinum, reach its peak development at puberty, and it has dual embryonic origin from ectoderm and mesoderm. It has a connective tissue capsule that penetrates deeply and divided the parenchyma into incomplete lobules. That means there is a joining region between adjacent lobules. Each lobe has a peripheral dark zone called the cortex and the central light area called the medulla. In this section, we can see the lobulation of the thymus tissue, and here we have the cortical region, the cortex dense region, and lighter stain area, the medulla. The thymic cells, we have a thymocyte, which are T cells precursor that found mainly in the cortex and reduced in the medulla. In addition, we have supporting cells called stellate epithelio-reticular cells that support the thymic tissue and joined by desmosome. Also, we have a dendritic reticulum cells found in the medulla. Thymic nerve cells, which are a subpopulation of epithelio-reticular cells found specifically in the cortex. And we have a thymic hustle corpuscle, which is a lamellated body, keratinized, eosinophilic body found in older age group, and it's found in the medullary region. In this section, we can see the epithelio-reticular cells that send septa that connected by desmosome and act as a support network for other thymic cells. Thymic hustle corpuscle, it is a lamellated body, eosinophilic. It's found in the medullary region. Vascularization of the thymus. We have a structure called the thymic blood barrier. since the thymus is responsible for production of T cells and they are formed in the thymus, so we have a barrier separated from the tissue of the thymic tissue itself. So we have no afferent lymphatics, so no lymph-filtration occur in the thymus. Efferent lymphatics leaves carrying the T cell to other organs. Thymic function is a site of terminal differentiation and selection of T lymphocytes, and it was found in research that neonatal removal of the thymus lead to partial or underdevelopment of secondary lymphoid organ and enable of the patient to respond to various antigens. Before birth, it is a primary lymphoid organ that receives stem cells from the bone marrow, then send T cells to blood and secondary organs. The self-reactive lymphocytes are selected and die by apoptosis, which is a programmed cell death, and they are phagocytes. So the self-reactive lymphocytes are eliminated, while the surviving lymphocytes migrate from the cortex to the medullary to be lived through the efferent lymphatics as T cells. At puberty, there will be reduction in the function of the thymic tissue, and this is called involution of the thymus, but still maintaining a low level of T cell production through the development for a mature precursor. So we have a picture of thymus in the involution state. We have replacement of lymphoid tissue by adipose tissue, and there will be still a little amount of normal lymphoid tissue that will be able to survive the T cell production through puberty. The spleen is the largest lymphoid tissue in the body, and it is interposed in the blood circulation rather than in the lymphatic circulation. It is important for blood filtration, so it is a site for antibody formation for blood-borne antigen, and it's a site for destruction of old RBCs. Generally, it is covered by a connective tissue capsule, and we have a trabecular that carry vascular structure called splenic vessels that entering through the hilum, and the exit of the certain structure includes veins and lymphatics also leave through the hilum. Splenic composed of reticular connective tissue containing reticular cells, lymphocytes, blood cells, macrophages, and antigen presenting cells. The splenic pulp parenchyma is divided into white and red pulp. In this figure, we can recognize the capsule of connective tissue, and these are the trabeculae sent from the capsule, and we have the parenchyma divided into white pulp, which is rich in the lymphoid tissue, and we have a red pulp. The red pulp made by a cord of cells called Burrow cord and surrounding sinusoidal channels, and it is rich with blood, so it is called red pulp. It contains T and B lymphocyte, macrophages, plasma cells, platelet, granulocyte, and red blood cells. In this figure, we can see this is the red pulp, and this side of bluish discoloration is the side of a white pulp. So we have the white pulp, and this is the red pulp that's filled with sinusoid. The white pulp made by dense lymphoid tissue called PALS, which is periarterial lymphoid sheath, sheathing an artery called the central artery. The central artery, although it's called central, it's like eccentric in position, so it is as a misnomer since it's located eccentrically. It is an arteriole rather than an artery, and it's subdivided into peninsular arterioles outside the white pulp. The marginal zone, which is a region surrounding the lymphoid nodule within the white pulp, containing blood sinusoid and loose lymphoid tissue and few lymphocytes and many active macrophages. In this figure, we can see this is the red pulp, and this is the white pulp of the spleen, and this is the central artery, which is eccentrically located, and it is an arteriole. The number 1 in the figure represents the germinal center that develop within the white pulp of the spleen. Splenic blood flow, we have two types of splenic blood flow. First, the splenic artery enter at the hilum and give to trabecular arteries that found within the trabeculae. Then we have central